Okay, good morning students. Today you got to see the next topic 1.6 post fertilization structure and events. In the last class we studied something about the fertilization process and what are the types of style and how the pollen tubes are entered inside this ovule. We already studied. The next another topic is post fertilization structure and events. In which way the fertilization is over, then what are the parts mostly changed into the next one or next another parts? We will see one by one. After fertilization, several changes takes place in the floral parts. There is a number of changes it will occur in the floral parts up to the formation of seed, the events after fertilization. Endosperm, embryo development, formation of seed, fruits are called post-fertilization changes. Here there is some of the changes are there. The sepals, petals and androecium, gynoeciums are there. That the parts fertilized, the androecium, gynoecium, pollination is occur, then fertilization is totally occur means what are the parts totally changed into, what are the ways. So you have to see. Here parts before fertilization, transformation after fertilization is there. There is a one table is there. Then what are the parts mostly present in a before fertilization? After fertilization, how they are changed? Sepals, petals and stamens, style and stigma usually wither and fall off. They are withered in a flower parts. The sepals and petals most probably. Then ovary it modified into fruits. Then ovule it modified into or changed into seed. Then egg is changed into zygote after fertilization. Then funicle, stalk of the seed, it will modified into stalk of the seed. Then micropyle ovule, there is micropyle of the seed, facilitates oxygen and water uptake. Because of there is a pore, nucellus perisperm, changed into perisperm. Outer integument of ovule, testa, there is a outer seed coat of outer seed coat, testa. The inner integument, there is a tegmen, there is inner seed coat. It is modified into tegmen, that is called inner seed coat. Synergid cells degenerate. Synergid cells are degenerate. Secondary nucleus endosperm, it is modified into endosperm. Antipodal cells are degenerate. These are disintegrate. Okay, these are the changes it will occur in a post-fertilization changes. After fertilization, the changes it will occur. The next one is endosperm. The primary endosperm nucleus, that is called PEN, primary endosperm nucleus. The primary endosperm nucleus divides immediately after fertilization, but before the zygote start to divide to form the endosperm. That is called endosperm. Here the primary endosperm nucleus is the result of triple fusion. How they are formed? The primary endosperm nucleus is the result of triple fusion, two polar nuclei and one sperm nucleus, and thus has 3n number of chromosomes are there. Here primary endosperm nucleus is the result of triple fusion. It will form because of two polar nuclei and one sperm nucleus joined together or fused together to form 3n number of chromosomes. Then in a nutritive tissue and a regulatory structure that nourishes the development embryo. Depending upon the mode of development, three types of endosperms are recognized in angiosperms. First one is nuclear endosperm. Second one is cellular endosperm. Third one is halobial endosperm. Nuclear endosperm, cellular endosperm, halobial endosperm. Then nuclear endosperm. First one, nuclear endosperm. Primary endosperm nucleus undergoes several mitotic division. The primary endosperm nucleus, PEN, undergoes several mitotic division without cell wall formation. Underline that word, without cell wall formation. Thus, free nuclear condition exists in the endosperm. For example, Coccinia, Capsula and Arachis. Then next one is cellular endosperm. The primary endosperm nucleus divides into two nuclei and it is immediately followed by wall formation. Here the wall formation it will occur. Subsequent divisions also follow cell wall formation. Example, Addaxia, Helianthus and Scoparia. The next one is third one is halobial endosperm. Primary endosperm nucleus move towards base of embryo sac and divides into two nuclei. Primary endosperm nucleus PEN moves towards the base of the embryo sacs and divides into two nuclei. Cell wall formation also occur, leading to the formation of large micropylar and small chala cell chamber end. Cell wall formation also takes place, leading to the formation of large micropyle and small chala cell chamber. The nucleus of the micropylar chamber undergoes several free nuclear division, whereas that of chala cell chamber may or may not be divided. Example, hydrilla and Wallisneria. These are the three types of endosperm. There is nuclear endosperm, cellular endosperm, heliobale endosperm. There is a diagram of that one. There is a nuclear endosperm, free nuclei is there. There is no cell wall formation, it will occur. There is several mitotic divisions as it will occur. Here, in a cellular endosperm, cell wall formation, it will occur in a cellular endosperm. After that, there is a heliobale endosperm. Here also, there is a mitotic division also occur, and also the cell wall formation, it will occur in this region. Okay. Then, here there is the same way. The endosperms may either be completely consumed by the developing embryo, or it may persist in the mature seeds. Those seeds without endosperms are called non-endospermous or X-albuminous seeds. X-albuminous seeds. You got to underline that word. Which one is non-endospermous or X-albuminous seeds? The seeds without endosperms. The seeds without endosperms are called non-endospermous or X-albuminous seeds. Example: pea, groundnut, and beans. Those seeds with the endosperms are called endospermous or albuminous seeds. They have endosperms. The seeds have endosperms. The endosperms in these seeds supply nutrition to the embryo during seed germination. Example: paddy, coconut, and castor. Okay, the next another important endosperm. Another apart from this pea endosperm, another one is there, that is called ruminate endosperm. Ruminate endosperm. The endosperm with irregularly and unevenness. The endosperms are irregularly present and unevenness in its surface forms. Ruminate endosperm, that is called ruminate endosperm. Example: Erica cachu and Passiflora and Myristica. So this is the ruminate endosperm. The next one, what are the functions of endosperm? What are the functions it will present in an endosperm? It is a nutritive tissue for the developing embryo. It gives the nutritive value for the developing embryo. In majority of angiosperm, the zygote divides only after the development of endosperm. In majority of angiosperm, the digotes divides only after the development of endosperm. Okay, endosperm regulates the precise mode of embryo development. Endosperm regulates the precise mode of embryo development. Then next another topic is development of dicot embryo. Up to this one you got to study what do you mean by the post-fertilization changes. There is one type of question. Another one is what is endosperm, what are the types of endosperm, what are the functions of endosperm. The next one is development of dicot embryo. Here how the embryo is formed, the stages involved in the development of dicot embryo. It is mostly referred Capsella bursa postoris, Onagrad or crucifix type is given in figure. The stages mostly involved in the development of dicot embryo. Here the embryo development at the micropylar end of embryo sac. First of all, it developed from the micropylar end of embryo sac. The zygote undergoes transverse division to form upper or terminal cells and lower or basal cells. The zygote undergoes some of the transverse division to form upper or terminal cell and lower or basal cells. You can see the diagram of the embryo development. There is a zygote. It go for two-celled proembryo. There is first one is terminal cell, below one is basal cell. These cells are again divided into four-celled proembryo. There is upper region. There are two cells again basal cells also two cells are there. After that there is a globular cells, a globular embryo it will present. There is embryonal mass also present. Hypophytes is also there. Then also suspensor cells also present in this region. Then heart-shaped cells are there also present. Hypophysis, embryonal masses also present. Suspensor cells also there. Cotyledon, then hypophysis, radical, root cap, suspensor cells also there. Then plumule it will present. Cotyledons also present in this region. then radicle and root cap. Radicle also present and plumule also there. Plumule is the upper region of this embryo. Radicle is the lower region, that is root region. Okay, further division in a zygote during the development lead to formation of embryo. Embryo undergo globular heart-shaped stages before reaching a mature stage. Embryo undergoes, there is some of the division. It go for globular heart-shaped stages before reaching a mature stage. The mature embryo has a radicle, two cotyledons and a plumule. Then seed. The fertilized ovule is called seed and possesses an embryo. Endosperm and the protective coat. Endosperm and a protective coat. The fertilized ovule is called seed and possesses an embryo, endosperm and a protective coat. These are called totally a seed. Seeds may be endospermous, that is wheat, maize, barley or sunflower, or non-endospermous, anything is there. Bean, mango, orchids and cucurbits also there. Then with the help of the one example, you can see sisal seed, example of dicot seed, sisal seed. With the help of this sisal seed, you can describe the dicot seed. The mature seeds are attached to the fruit wall by a stalk called funiculus. But underline that one, which one is funiculus? The mature seeds are attached. Mature seeds are attached to the fruit wall by a stalk is called funiculus. The funiculus disappears, leaving a scar called hilum. The funiculus disappears, leaving a scar called hilum. Below the hilum, a small pore called micropyle is present. Below the hilum region, a small pore is present, that is called micropyle. With the help of this micropyle, the water and oxygen also enter inside this one. It facilitates entry of oxygen and water into the seeds during germination. During the germination period only, the water and oxygen are enter inside this micropylar region because it is a pore-like structure it will present. Each seed has a thick outer covering called seed coat. There is a coat it will present, that is called seed coat. There is a thick outer covering. The seed coat is developed from indegments of the ovule. The outer coat is called testa, already we studied testa, and is called hard, whereas the inner coat is thin in nature, membranous, and it is called tegmen. Outer coat is testa, inner coat is called tegmen. Inner coat is very thin in nature, outer coat is very hard in nature. In pea plant, the tegmen and testa are fused together. In a pea plant, only the tegma and testa, these are fused together. Two cotyledons laterally attach to the embryonic axis and store the food material in pea, whereas in other seeds like castor, the endosperms contain reserve food materials and cotyledons are very thin. The portion of embryonal axis projecting embryo. Portion of embryonal axis projecting beyond the cotyledon is called a radicle or embryonic root. You can underline that word radicle, and which one is radicle and embryonic root. There is a projecting beyond the cotyledon. Projection beyond the cotyledon is called radicle. Or the other end of the axis is called embryonic shoot. There is other end. It will present above the cotyledon region. The other end of the axis called embryonic shoot, that is called plumule. The embryonal axis above the level of cotyledon is called epicotyle. And the embryonal axis above the cotyledon is called epicotyle. Epi means above the cotyledon region, whereas the cylindrical region between the level of cotyledon, a cylindrical region below the region of between the level of cotyledon is called hypocotyle. Below the cotyledon region, that is called hypocotyledon. Hypo means nothing but below. Here you can see the diagram of the Sisal side. There is testa, there is the outer coat, radicle, and testa, there is the outer coat. Tegmen is inner coat, sheet entire is there. Radicle, it will present. Radicle, there is a root system. Plumule is the upper part of this region, it will grow the stem region, shoot region. There is a plumule, it will grow the shoot. Cotyledon, these are two cotyledon. Above the cotyledon region, that is called epicotyle. Below the cotyledon region, that is called hypocotyle. Radicle is there, testa is there, seed cut opened. The next one is a oryza seed, example for monocot seeds. In this seed, the seed of paddy is one seeded and it is called caryopsis. Underline that one, caryopsis. It only mentioned in a a single monocot seeds only. The seed of paddy is one seeded and it is called caryopsis. Each seed remains enclosed by a brownish husk region which consists of glyms around in two rows. The seed coat is a brownish membranous layer closely adhered to the grain. Endosperms form the bulk of the grain and it is storage tissue. It is separated from the embryo by a definite layer called epithelium. Underline that one. Which one is epithelium? Here it is separated. It is separated from the embryo by a definite layer called epithelium. The embryo is small and consists of one shield shape. You have to underline. Very important. The embryo is very, very small and consists of one shield shape. One shield shape, that is one shield shape cotyledon is called scutellum. Scutellum it will present only in a monocot plants towards the lateral side of embryonal axis. It will present towards the lateral side of embryonal axis. Okay, the short axis with the plumule and radicle protected by the root cap. It's very important. Root cap is present in all the seed. All the plant, the root cap is present. Root axis with the plumule and radicle protected by the root cap is present. The plumule is surrounded by a protective sheath called coleoptile. The plumule is surrounded by a protective sheath is called coleoptile. The radicle including root cap is also covered by a protective sheath is called coleorhizates. Rhizates means nothing but roots. Rhizates are nothing but roots. Coleorhizates and coleoptile. The scutellum supplies the growing embryo. The scutellum supplies the growing embryo with the food material absorbed from the endosperm with the help of the epithelium cells. Okay, you can see the diagram of that one. Fruit caryopsis, entire caryopsis there. Husk region is there. Endosperm, scutellum is present. Coleoptile, shoot apex. There is an innermost region of this one, and also radicle. It will grow the root and the root cap. Coleorhizates also there, LS of fruits also there. Okay. Up to this one you have to read very carefully. The next class we will see the apomixis. Today classes we have to see what are the post-fertilization changes and what are the types of endosperm. What is endosperm? Types of endosperm and functions of endosperm and development of embryo and dicot seeds and monocot seeds. That's all. Dicot seeds, isocete seeds and monocot seeds you can study paddy, caryopsis. Okay, thank you one and all. Thank you.